students, we are proceeding looking on the biochemical tests of carbohydrate, and we have seen that most of the carbohydrate, they are reducing sugar. They are reducing sugar due to the carbonyl group that are present in their structure. And that carbonyl group, we have seen that the first one is the aldehyde and the second one is the ketones, and thus bring about a reducing nature within the carbohydrate compound. So now let's proceed. We are saying that the carbohydrate may be present as isolated molecule, or they may be physically isolated or chemically bound to other molecule. So hizi carbohydrate unaweza ukaikuta iko yenyewe, au unaweza ukaikuta carbohydrate combined with the other food substances. For instance, unaweza ukakuta carbohydrate imo ndani ya protein. Au unaweza ukaikuta carbohydrate it is chemically combined with the other molecules. So how can you identify there is a presence of carbohydrate? And here now we can simply say that the specific test for a particular type of carbohydrate is based on the presence of specific component of carbohydrate or the reducing property resulting from aldehyde or ketone. Kwa unaweza uka-identify there is a particular carbohydrate is due to the presence of specific component of carbohydrate that compose of the carbonyl group, that is the aldehyde and the ketone. Is where now you can find that there is a presence of a carbohydrate. So now we can say that different method can be used to test for the presence of the starch reducing sugar and non-reducing sugar. So different methods are being used. So we have a lot of method. This method ndizo ambazo sasa zinatumika ili kwenda kujua kwenye mchanganyiko fulani if there is a presence of carbohydrate. And here we are going to test only 3 things in carbohydrate. The first, we are testing in the presence of starch, and also we are testing the presence of reducing sugar, and also we are going to test the presence of non-reducing sugar. If you will notice, there is a presence of either of 3 of these carbohydrate, meaning that there is a presence of carbohydrate into your chemical. So this table 1.3, we're going to see it later on, while we are going to discuss the structure of the basis of taste of carbohydrate. And now let's start by looking on the biochemical test for starch. So how can we test the presence of starch in any solution? So here we are saying that the presence of starch is a biological material can be tested by using iodine solution. So the use of iodine to test the presence of starch is one of the common experiments. So this experiment, it is well known. Kwa maana kwamba we used to test if there is a presence of starch by using this chemical, that is the iodine solution. So we just take our solution, then we add drops of iodine solution, then we detect if there is a presence of starch, it will tend to change its color. So now we can say that a solution of iodine, that is iodine molecule, and potassium iodide in water has light orange brown color. So this is the chemical which is used to test the presence of starch. Kwa maana kwamba mwonekano wake unakuwa na this color, that is light orange brown color. So if it is added to a sample that contains starch, the color will change to a dark blue or blue black. So ukichukua ule mchanganyiko wa iodine molecule together with the potassium iodide, ukaja ukachanganya kwenye your sample into any sample, is whereby your sample now will change its color to dark blue or blue black. So the presence of blue-black, meaning that into your sample or into your solution, there is a presence of carbohydrate, that is a starch. And we can say that in the absence of starch, the orange-brown color of the aqueous solution remain. Kwa maana kwamba pale ambapo utakuta kwamba hakuna the presence of the starch, your sample or your sample solution will remain its color, that is the orange brown, that is orange brown color, which is the color of the mixture of your chemical, that is iodine molecule and the potassium iodide. So kibakia ile rangi ya ile reagent yako ambayo umeitumia kuiweka mule, kibaki ile ujue kwamba hapo hakuna the presence of starch. So that is how we are going to test. So the basis of test now, how can we test? The basis of test. We say that the iodine bound inside the helical structure of the amylose forms a dark blue color. So how is it occurring? Tunasema kwamba the basis of test, that is the iodine bound into the helical structure of the amylose, form a dark blue color or blue-black. Kwa maana kwamba kivipi sasa mpaka ile dark blue unaiona pale kwenye your sample? Unaiona pale kwa sababu kwenye ile starch, kwenye ile structure of starch tumesema imetengenezwa na amylose pamoja na amylopectin. So ile amylose ambayo imezunguka ile starch ndio ambayo sasa ita unite au itaungana na ile iodine kutengeneza ile dark blue ambayo sasa wewe utakuja kuiobserve as there is a presence of starch. So the reaction is due to the formation of polyiodide chain, that is complex, from the reaction of the starch and the iodide. So the dark blue color, which you can be sure, or you can see or you can observe, is due to the reaction or the combination between the starch and the iodine. And remember, the part of a starch which is responsible to combine with the iodine, it is known as the amylose. And now here we can say that the amylose in the starch form the helical where iodine molecule are assembled to form a dark blue color. Kwa hiyo sasa ile ina form a helical structure within the sample or within the solution. You cannot see all of this process by using your naked eyes until you are going to use the microscope. So you're going to observe all of this. So here we are talking about that amylose, which is the structure of the starch. So ile utakuja kuiona pale ambapo sasa ile amylose itakuwa imeungana na ile iodine. Kwa maana kwamba ile amylose ina create mazingira ya ile iodine kuja kujishikiza ndani ya ile amylose na kutengeneza hiyo color. So now let's see. Now you can see amylose in the form, amylose in the starch forms the helix. So you're going to see this helix where iodine molecules are assembled or forming dark blue color. That is, let's see this figure. So this is what we are going to talk about. That is a helical structure of amylose bound the iodine molecule. So this one, all of this structure, it is the amylose. But this one, it seems like a blue, or this one is the dark blue. This is iodine. This is iodine complex, that is the iodine ion. So ikija ikaungana na hii amylose structure, ndipo ambapo sasa itakuja kutengeneza hiyo dark blue color ambayo sasa unaweza ukaiona au ukaiobserve kwenye ile experiment yako. So that is how it is formed. And my student, by saying so, this marks the end of our today's session. So make sure that you are following the next session, because there we are going to clarify also more about the basis of test of reducing sugar, non-reducing sugar, and other molecule or organic molecule. Thank you and for following us at Educare TZ. Bye bye. Have a nice moment.